Hello lovelies, in this video Brilliant Dr. Redwoods is going to be taking you through all of the different plant hormones that you need to be aware of for your A-level biology.
Plant hormones. Plant growth is controlled by different plant hormones. Growth in plants only happens in meristem tissue, so these are cells that have the ability to divide, much like stem cells in animals. There are a few different meristem locations in a plant shoot. Tips of roots and shoots are known as apical meristems. This is how the shoot tips and the root tips elongate, how they get longer. There are cylindrical lateral meristems around the edges of roots and shoots, and this allows both roots and shoots to get wider. The lateral bud meristems give rise to extra side shoots, and then the intercalary meristem, which is located between nodes, which is where the leaves and the buds branch off. The meristem tissue between them helps the shoot to elongate as well between the nodes. So the hormones that control growth at these meristems are produced in certain tissues, and then they will travel to their target tissues. Plant hormones can travel by diffusion from cell to cell, active transport as well, or by mass flow in either the xylem or the phloem. The hormones can influence cell division, cell elongation, or cell differentiation, and all of these obviously involve different ways of either growing, producing more cells, or turning those cells into specialized cells. And they can work together, so some hormones can work together to achieve certain growth measurements in plants, and they can be also, some of them, antagonistic to each other. So some hormones may inhibit the effects of others. So the first group of hormones we're going to look at is auxins. So auxins are plant hormones that are produced in the apex of roots and shoots that regulate plant growth. So we've already looked at phototropism and geotropism and how auxins regulate the growth of cells by causing them to elongate or inhibiting elongation. In this example, we're going to look at how auxins have been shown to inhibit the growth of lateral buds. This inhibition of the growth of side shoots by the apex or the apical meristem is known as apical dominance. We need to be able to talk through and explain the evidence that was used to support this theory that it's the auxins in the tip that are causing the inhibition of these side shoot growth. So the first piece of evidence was that lateral buds did not grow when auxin paste was applied to a cut apex. So we've cut the tip, removed the apical meristem, and then if auxin paste was applied, lateral shoots still didn't grow. So when auxin transport from the apex was inhibited, so we blocked any auxin from being able to move down the shoot, then the lateral buds grew. So in the first case, we removed the source of auxin, but we applied auxin paste. That stopped the lateral buds from growing. So that suggests it is the auxin. Then it was like, oh, is it just not being transported down? How do you know? Well, by blocking auxin transport, that caused those shoots to be able to grow. So that is another piece of evidence to support it. And what happened was that they removed the apex and they would measure the auxin production at the lateral buds. And actually, auxin production increased at the lateral buds when we didn't have any source of auxin at the top. And that's what caused their growth. So we know that this creates these increased concentrations of auxin around the lateral buds, and that causes it to grow when we remove the shoot tip. But we do know that abscisic acid and cytokinins, two other plant hormone groups, have been shown to also affect apical dominance. So although this is very compelling evidence that auxin has something to do with the apical dominance and its presence in the apical meristem is something to do with inhibiting that lateral bud growth, we don't know for sure that it doesn't also involve these other two hormones. There was also a suggestion that when cutting the tip, oxygen was allowed to get into the top of the shoot, and that may have also affected the results. So with plant hormones, there's always got to be lots of evidence built up through different methods to try and demonstrate what's actually happening in terms of hormone movement and what causes it changing, to show that there's a causal link between these hormone changes or the actions you're doing to the shoot and what actual results it has. So let's look at how abscisic acid and cytokinins can potentially interact with auxins to look at bud growth. So as far as we know, ABA or abscisic acid inhibits bud growth, similar to auxin. This is because high auxin concentration promotes high abscisic acid levels. And the evidence to support this is that obviously when the apex is removed, the auxin levels decrease, and so the ABA levels also decrease. Cytokinins is another hormone group, and these can promote bud growth. Direct application, so actually like putting cytokinins onto these lateral buds, was shown to override the apical dominance. So even with the apex present, if you apply cytokinins to the bud, they will grow anyway. So what was found to be happening was that high auxin concentration causes most cytokinins to be sent to the apex. It creates a cytokinin sink in the apex. So that is where the majority of cytokinins in the plant travel to. But when the apex is removed, the cytokinins don't have that drive to go towards the apex anymore. So they spread out more evenly through the plant, and that's when they go to the lateral buds and they can promote that growth. These show that it may not be a direct link between one hormone and a change in the plant. It can be an interplay, an interaction between other hormones also causing the effects. The next hormone group we're going to look at is gibberellins. Gibberellins are known to control stem elongation and seed germination in plants. Gibberellic acid, or GA, when applied to dwarf or short plants, caused them to grow taller. So this was obviously the synthetic application of a hormone to the outside of plants. So that helps, that is a piece of evidence, but to prove that it works inside plants in the same way, that the naturally producing gibberellins actually cause this change, and the process or the mechanism that it does that was needed to be shown a bit more scientifically. So to investigate, they actually measured the concentration of gibberellic acid in different plants. So they found two varieties of plants, one that had a dominant allele, capital LE, and those that were tall had this dominant allele, so they were either homozygous dominant or heterozygous, compared to a dwarf variety that didn't have this dominant allele. And they found that these taller plants had higher concentrations of gibberellic acid, or GA1, compared to the dwarf plants. And what they discovered was that the dwarf plants and that homozygous recessive allele meant that they had no enzyme to convert GA20, a precursor of gibberellic acid, into GA1. And so that is what the dominant allele provided in those taller plants. They also found another type of mutation in plants that meant that they didn't produce gibberellins at all, and these plants only were able to grow to about 1 centimetre tall. This is because they weren't able to produce the GA20, the precursor to GA1. So in order to determine that it was definitely the GA1 that was causing this elongation and growth in the tall plants, they grafted, which means they literally took the top of one plant and sort of attached it to the bottom of another plant, and they will kind of grow together and form a new plant made of two halves. So they grafted a dwarf homozygous recessive shoot, which has no enzyme to make GA1, but does have the precursor GA20, and they grafted that onto the roots of a plant that had the mutation that had no GA20 but does have the enzyme to produce the GA1 from the GA20. So what they managed to do was graft these two together. So take our homozygous recessive dwarf plant, the shoot, and graft it onto the mutant that has the enzyme, so it's not homozygous recessive dwarf in the same way, but it just has a different mutation that means it doesn't produce GA20. Put that on the bottom, and then what happened was, what they observed was that that shoot actually then was able to grow really tall. So it was no longer a dwarf plant. So because they had access to both the GA20 now and the enzyme to convert that GA20 into the GA1, therefore it grew tall. So there we go. There is my tall plant that is actually a dwarf shoot, which would have homozygous recessive and not have the ability to make GA1, and my rootstock that does have the enzyme to make GA1 but doesn't have the ability to do it because it doesn't have GA20. And then we put those together, and so we've got the enzyme and the substrate, and we're able to make a tall plant. So clearly the gibberellic acid actually caused that elongated growth. Hormones can also control other plant responses that are not directly linked to growth. Leaf loss or abscission is controlled by auxins and ethene. We know that a lot of deciduous plants lose their leaves in winter, and this is to help conserve water loss. Auxins inhibit leaf loss in young leaves, but this amount of auxin reduces as those leaves age. Ethene stimulates leaf loss as it's produced in older leaves. Ethene causes leaf loss by causing an abscission layer of cells to form between the leaf and the stem. It then causes this band or layer of cells to expand, which breaks the cell walls and the leaf falls. Again, there is an interaction here with cytokinins, so we can get an interaction between the cytokinins, the auxins, and the ethene, because cytokinins can actually kind of inhibit this and delay leaf senescence or leaf drop, which can be useful in some industries to try and prevent that. One example is to try and prevent the yellowing of leaves like lettuce once they've been cut and ready to be sold. Stomatal closure is also controlled by abscisic acid, or ABA. When a plant is suffering from water stress, like drought, ABA binds to receptors on guard cells, causing calcium ion channels to open. Calcium ions enter, and they cause other ion channels to open in the guard cell, and these ions then diffuse out. The loss of ions increases the water potential of the guard cell, and it decreases the water potential surrounding the cell. So water follows the ions and leaves by osmosis. This causes the guard cells to become flaccid and causes them to close.
Ouch! This is why in some videos I'll explain scratches.